And welcome back to ATTV. I am here with Astrea Bioseparations today. Really excited to have a conversation with you.
Thank you.
Would like to introduce yourself?
Yeah, so my name's Ian Scanlon. I'm the fiber development lead here at, Astrea Bioseparations. We're based in Cambridge in the UK, and we've been working on this new material now for a couple of years.
Excellent. So. And I'm Ben McLeod, your host, founder of Convey Bio. Would love to get your thoughts on kind of what you see as the trends inside of the industry. What do you think people should be focusing on?
So I think that what we're seeing at the conference today, and a lot of the information that people are presenting, there's such a big variety. Like there's obviously you get your cell therapies and things going through approval very very quickly. There's more and more of that growing. But I think really the interesting thing is the sheer kind of diversity in the portfolios that are coming through. And we're seeing sort of some things will succeed, some things won't. But I think if you look at the way that people are moving, say from particularly in the Lenti world, from pure kind of the cell therapy applications towards in vivo material that they're looking for, going directly into the patients and looking at the novelty or the novel versions of sort of plasmid direct design that's coming through. I think there's a lot of energy in the space at the moment, and it's really interesting to see that keep growing.
Excellent, very good. So let's dive in a little bit about Astrea and what you guys are working on. How are you supporting the cell and gene therapy sector?
So we decided a couple of years ago really to focus with the development of our new material, the real key driver for the design sort of philosophy initially was to take something from scratch and design it for these new vectors. So the things have been around for, I've been working on lentivirus for 25 years, right? But there's not really been anything like specifically built from the ground up for that. So with our material, we've got our copolymer that we can electrospin, and we've designed that to have a really large pore size, to be flexible, to be stable to the kind of sanitization processes that we need, and really to build it with a kind of end user in mind. So we have our one offering that's for like the plasmid market, where we're looking at, that's a key component, sort of critical intermediate for every vector process. And then what we decided was like lentivirus is the thing that we thought was least well served by the current process. You've got a lot of strong anion exchanges, so you have to then elute at high salt, and then that damages the efficacy of your lentivirus. And you've also got the kind of the traditional resins are not really fit for purpose for those kind of really large targets, and that's how we want to help serve that market.
Excellent. So let's dive in a little bit more on that. So particularly with lentivirus purification, what are some of the main challenges with it?
So it's really hard. It is one of the big problems. So the big issue that when we started kind of looking at this, from the background of we had looked at current processes, and you're looking at highly variable upstream processes. You get huge variation when you move between different GOIs. That can affect the expression quite drastically. You can then have you're hitting the front end of a process where if you've got a strong anion exchanger or the kind of processes that currently are around there, that's if you have got a bind dilute process, because a lot of the existing ones were derived from sort of centrifugation or TFF, where you're separating out some of the smaller sort of impurities, but nothing that's the same size. Right. And we were experiencing issues with recovery in common with everybody else that does that. So capacity and recovery, they're the classic downstream challenges. It's kind of the same as where people were with MABs in the beginning of those processes. You need to have a process where you can recover the most functional material that you possibly can. And the fragility of the lentivirus is your enemy there. But additionally, there are other sort of challenges around it, which is you've got to remove things that are very closely related. You've got DNA that's very closely charge related. You've got potentially other component like non-functional particles that you have to remove. So we want to provide a platform, but we also want to have enough kind of flexibility within that operational kind of parameters that people can optimize for their process, which we think we are able to give them.
Very good. So yeah, you've outlined some of the challenges here, and maybe we can just recap. How is Astrea helping with those challenges?
Yeah, so I think
it being really, really hard.
It is hard. Let's not soft soap it. It's really hard, right? But I think that the main approach that we took was around looking at a ligand where we thought that we wanted to give ourselves more flexibility. So we went for the weak anion exchanger. You've got pH there, then you've got pH and salt that you can play with for the elution. And also we're looking at, like, we've got such a high surface area for our electrospun fiber. You know, it is basically a network of kind of threads of material. So it has this huge surface area. So you don't need to go to very high ligand density to get high capacity.
Right.
So you can elute effectively at lower salt conditions, even if you are like operating at the same pH. So we've shown that quite nicely. We're showing some data in a couple of months at a month and a half at ACS around that. But we have specifically looked at that for the Lenti process, so that we can get better functional recovery, lower salt, then you affect the further downstream formulation process, because you don't need to dilute as much. So you've got a lower volume to process for your TFF, and that also makes your filtration recovery better. So final filtration and removal of any kind of bioburden is also a big challenge for these kind of processes. And we looked at that. Can we keep the footprint down? Can we keep the conditions as mild as we possibly can? And we think we've got some good data showing we can get good recovery, and we're constantly improving it as well. So we're looking at other options for elution salts, other kind of conditions, all of which our team are really focused on the entire process. We have some really great chemists and people and material scientists looking at material. We've also got a whole team focused on generating the lenti and purifying it, so we know what the customers are trying to do.
Gotcha. So in terms of lentivirus purification, which, as you've said, is very, very hard, how does your approach differ from other methods that are out there?
So I think if you look at a lot of the traditional processes, like I said, 20 odd years ago when we were looking at this, you would just do centrifugation, or you do SEC, where you can remove everything based on the principles of, just you're moving all these small impurities. But that doesn't give you any kind of concentration unless you then concentrate with your TFF. But if you concentrate your TFF before you've purified it, you're going to get a lot of problems with aggregation, and we see that particularly with like host cell DNA contamination. Right. And I think that having a bind dilute is for me really key in terms of keeping the footprint of the process down and making sure that you're providing yourself the best chance of getting that kind of functional particle at the end. And we really focused on how can we then take that and put in an anion exchange ligand that will give us that flexibility to go across a lot of different pseudotypes, because as I say, people are moving into looking at different pseudotypes, not just your VSVG classic kind of CAR-T process. So potentially, if you've got an affinity where it's just for VSVG, that's not going to serve the entire market. And then a lot of the strong anion exchanges, which are all developed, they're basically the products that are used for Lenti are primarily designed for removing DNA from protein purification. So they may not have the best kind of optimized process for recovering what is bound to the surface, because ultimately that's not your interest. So we really wanted to build it with the idea that you're going to bind the Lenti and you're going to recover it, and it's going to be functional when you recover it. So we took it from first principles.
Very good. And so in terms of process intensification, how does this help?
So I think we see a lot of opportunities there. So again, if you're running a kind of conventional resin-based process, because of the reduced capacity that you get from not being able to diffuse into the beads, like with your big particles or your big plasmid DNA, then you can't run very fast. So if you've got an adsorbent like ours where you can run at very low resonance times because of your very high surface area, then you can have a continuous process. You can kind of get your process intensification by either like just shrinking process time, or more generally as having maybe a continuous process that runs over a longer period but gives you that consistent elution of the product. So we're really interested in the application of that with, say, like simulated moving bed for like continuous capture of different particles or different targets, and whether the advantage of having this very open surface, which would, if you're overloading it, you get even better kind of utilization of the entire surface area.
Right.
So we think that potentially that's one way that we can really supercharge the kind of process of intensification process. But also just looking at it from a more broad application of, if you can elute at lower salt, you dilute less, you end up with less. Your pool at the end is smaller, so therefore you can get more material out per unit volume of your TFF at the end. So we're hoping to be able to help people in various points of the process.
Excellent. And then in terms of future plans, what's next?
So always get asked about kind of going bigger, and that's something that we're like. The manufacturing process is a semi-continuous process. You can electrospin onto it. It looks like a roll of wallpaper, effectively. When you get it, you get your roll of wallpaper. You've got that. You can make it as long as you want. So there's no kind of notional sort of ceiling on how much of the material itself we can make. Then it's more of a question of, what does the market need? So do we need to make a 1 liter device when you can use the 100 ml device to process 50 liters of lentivirus? Maybe not, right? Because maybe that's big enough, or maybe 500 ml would be big enough. But then when we think about the plasmid market, where you can go up to sort of much larger expression levels for, say, like vaccine purification, we're looking at producing a kind of 1 liter scale version of that. And then there's diversification in the material itself. I like to think of it as where we are now is where sort of agarose resins were in, say, like the 50s, right? This is the first commercially available materials now with these electrospun fibers. There are lots of other things we can do. The process itself, where we co-spin the material, is quite flexible. So we could put lots of different, if you can dissolve it, and we try and dissolve everything in aqueous phases, so that we're not looking at any kind of hazardous waste streams or difficult to handle materials. But we have found we can spin functionality into the fibers themselves, and therefore then you cut down on the chemical processing. There are other materials that we could look at, other ligands, obviously, that we could look at. There are, sadly, we don't own any of those. The ones that people are very interested in asking me about routinely. But there's tremendous work being done with affinity tags for all sorts of different new targets. And if you think about just the flexibility of being able to attach them to the fiber, there's a lot of things that you could do in the future. We also want to go to the smaller scale to provide people with a kind of screening tool as well, or tools so particularly where you look at the extracellular vesicles kind of portion of the market, where the scale of operation is actually quite a lot smaller. We're looking at trying to serve that with something as well. So both ends. We're going to have a little one and a really big one, right? So
excellent. Sounds really good. So how can people get in contact with you here at the show, or outside of the show?
Well, outside of the show, I'd rather people didn't come up to me on the street, but like they can if they need to. So we have a booth over in the corner. I don't know if I can't remember what number it is at the moment, but it is 611. Okay, so there
611.
611, and we have our commercial team are there, always ready to take your call. And I think we'll be at ACS in March, and we have plans to keep going at all these different conferences. Okay,
excellent, great to speak with you, Ian, and all the best with the rest of the show.
Thanks, Ben. Thank you very much. Thank you.